Welcome to today's Explainer. You know, if we're talking about the chapter on genome engineering, we literally have to start in 1973. That was the year everything changed. Stanley Cohen and Herbert Boyer didn't just pull off some neat lab trick. They successfully transplanted genes from one living organism to another. They essentially unlocked the code of life right then and there. It kicked off the entire era of genetic engineering. Everything we're diving into today, from curing genetic diseases to tracking viruses, it all stems from that single monumental breakthrough.
So, today we're unpacking genome technology and engineering. To really wrap our heads around this chapter, I want you to think of the genome less like a dusty old biology textbook and more like a software code base, right? Nature's been writing this code for billions of years, but now we actually have the tech to read that code, to write our own lines, and to execute it to get these incredible real-world results.
Okay, let's dive into this. Here's our roadmap for the explainer. First, mapping the genome. Second, reading the genetic code. Third, editing the blueprint. Fourth, understanding genomics. And finally, the power of proteins.
All right, starting off with Section 1, mapping the genome, or locating the code.
Imagine trying to use an instruction manual to build a human being, but there's no table of contents and absolutely zero page numbers. Well, that was our first major hurdle. Early on, we had to rely on genetic mapping. It's a rougher method that uses crossover frequency and centimorgans to estimate relative distance. It's kind of like knowing a house is, 3 blocks past the supermarket. Helpful, but not exact. To actually engineer a genome, we needed physical mapping. By using restriction enzyme sites, SSLP and STS, we could suddenly get exact, high-resolution locations. We finally had the molecular street address.
And a real game changer for creating those physical maps was RFLP, or Restriction Fragment Length Polymorphism. Think of restriction enzymes as highly specific molecular scissors. They scan the DNA and only make a cut when they hit a very specific sequence. When they do, the DNA breaks into fragments of different sizes. By separating those out and looking at the visual map they create, we can compare individuals. This is actually exactly how we trace hereditary diseases within families, letting us pinpoint the precise physical location of a disease-causing gene.
Moving right along to Section 2, reading the genetic code through high-throughput sequencing.
So once we actually knew where to look, the next step was reading the individual letters, the A's, C's, T's, and G's. And the speed at which this tech evolved is just staggering. First-generation sequencing was a multi-step, painstakingly slow process using cloning vectors. But then we made this massive leap to next-generation sequencing, using Illumina flow cells to read millions of fragments in parallel. And today, we're into the third generation, using nanopores to read single molecules of DNA in real time. It's wild.
Let's quickly break down how that next-generation Illumina sequencing actually works. Step 1. You fragment the DNA into small pieces. Step 2. You attach these little chemical handles, oligonucleotide adapters, to the ends. Step 3. You wash them over a flow cell and use bridge PCR amplification to create dense clusters. And finally, Step 4. You read the sequence using fluorescent base tags. As each uniquely tagged letter binds to the DNA, it flashes a specific color. So you've got a camera recording millions of tiny, flashing lights, reading the code all at once.
And this brilliantly illustrates where we're heading with third-generation nanopore sequencing. Picture a single strand of DNA being pulled through a microscopic pore. As it moves through, an electrical current passes across it. Because those A, C, T, and G bases are physically shaped differently, they each change that ionic current in their own unique way. The machine measures those minute changes and translates them into the genetic sequence on the fly. Real-time, single-molecule reading. No cloning, no light flashes, just pure electrical translation. Which brings us to Section 3. The climax of the chapter, really. Editing the blueprint, or genome engineering.
We've mapped the coordinates, we've read the letters, so now, how do we actually rewrite the code? Well, early on, scientists essentially hijacked a trick from nature. Our genomes are packed with transposons, which you probably know as jumping genes. They naturally move around using a cut-and-paste mechanism. Researchers figured out that by engineering a transposon to insert into a target sequence, they could disrupt the coding frame. It basically knocks out existing genes, which is a fantastic way to study exactly what happens when you purposely break a specific piece of the biological program.
Now, what's really interesting about this slide is the sheer elegance of CRISPR-Cas9. This is the absolute superstar of genome editing. It uses a guide RNA to find the exact DNA signature we want to target.
Once it locks on, the Cas9 protein makes a super precise double-strand break. From there, we can either let the cell repair it with errors to knock the gene out, or, and here's the real magic, we can provide a custom template to seamlessly rewrite that section of code. All right, moving into Section 4, understanding genomics and making sense of the data.
So we've got all these amazing tools, right? But what do we actually do with this massive mountain of data? That's where the different branches of genomics come in. Whole genome sequencing gives us the entire DNA map, which is huge for personalized medicine. Then you have structural genomics, looking at how DNA is physically organized in 3D. Functional genomics tells us how that genetic info is actually executed into physiological functions. And comparative genomics compares species to find core, essential genes versus the ones that make a species totally unique. It's how we see the big picture.
And finally, Section 5, the power of proteins, where we actually run the program.
So the crucial point is that editing the code isn't the end goal. DNA is just the blueprint. The proteins do the actual heavy lifting. By tweaking the DNA, we can engineer proteins with some wild applications. We're talking increased stability under extreme temperatures or pH levels. We're developing novel reagents for diagnostics. We can track exact protein locations within living cells, and absolutely most importantly, we're creating highly targeted therapeutics to fight complex diseases.
To really drive this home, look at these two examples. On the left, we have green fluorescent protein, or GFP, illuminating a cell nucleus. It essentially acts as a molecular flashlight for researchers. On the right, we have a recombinant immunotoxin binding to a cancer cell. This is a custom-built biological assassin. We've combined the precise targeting of an antibody with the lethal payload of a toxin. It hunts down specific cancer cells and destroys them, completely ignoring your healthy cells. Just incredible engineering.
From mapping rough genetic locations to watching single molecules of DNA fly through a nanopore, all the way to deploying custom-built proteins that literally hunt down disease, the journey in this chapter is breathtaking. We've turned biology into a programmable medium. We read the code, we edit the blueprint, we execute the program. So, I'll leave you with this. Now that we have the ultimate toolkit to rewrite the code of life, what are we going to choose to build next? Thanks for tuning into this explainer, and we'll catch you next time.